Hello everyone, this is Swarangi and Vidhi. Today we are going to present you about numerical taxonomy, the underlying mechanism and construction of the phylogenetic tree, along with its merits and demerits.
Numerical taxonomy, also known as the Adansonian taxonomy, is a technique of classifying microorganisms using numerical methods. Robert Sokal and Peter Sneath are the significantly responsible for the development and popularization of numerical taxonomy, also referred to as taximetrics or phonetics. It is the numerical evaluation of similarities or affinities of the taxonomic units, which are typically classified into taxas based on their affinities. In this type of taxonomical classification, individuals are chosen as well as their characteristics are spotted to construct the taxonomic category. Each characteristic individual, genus, or species is referred to as an operational taxonomic unit. Then, using character analysis, similarities between the individuals are determined.
The principles of numerical taxonomy are as follows. First, the greater the content of information in the taxa and the more the characters taken into consideration, the better a given classification system will be. Second, every character should be given equal weightage in creating a new taxa. Third, the overall similarity between any two entities is considered for the purpose of comparison. Fourth, the correlation of characters differs in the groups of organisms under study. Thus, distinct taxa can be recognized. Fifth, it does not necessarily reflect evolutionary family relationships. And sixth, the science of taxonomy is viewed and practiced as an empirical science.
To understand numerical taxonomy in a better way, we have taken 6 operational taxonomic units, that is, 6 different bacterial species of varying 10 characteristics which might be similar to some while different to others. Here are the 6 morphological characteristics of these bacterial species.
To study the numerical taxonomy, we have taken 6 different types of microorganisms, 6 different types of bacteria in specific. Every bacteria has its specific characteristics which can be studied. So, first here we have taken a bacilli which has a capsule and it is, as we can see, it is an endospore producing. And the pink region, that is the pink layer here, shows the presence of cell membrane, while the inner cotton layer shows the presence of thick peptidoglycan. This bacillus is has peritrichous flagella, while the second, which we have taken, is also a peritrichous flagella, but it has 3 layers of the outer covering, that is a cell membrane, a peptidoglycan layer and a cell wall, that is a lipopolysaccharide, which indicates it is a negative bacteria. And both of them were bacilli. Even the third one, which has a flagella, but it is a monoflagellated microorganism. With similar to the second one, it has 3 layers: the cell membrane, the thin peptidoglycan layer and the outer lipopolysaccharide layer. So, here are 2 cocobacilli of different width and length. Here we can see the width of this cocobacilli is bigger than this one, but along with it, it is a gram-positive microorganism by the presence of only 2 layers of outer covering. That is, it is a cell membrane, along with it a thick peptidoglycan layer. And it has no flagella, but has a capability of producing a capsule. Similarly, it also can produce a capsule but it is a gram-negative microorganism. Similarly, as we have seen earlier, that there are 3 layers of the outer membrane along with a capsule. This is a cocci which can be seen clearly, which is a positive cocci, as there is a thick layer of a peptidoglycan and a thin and a pink layer of the cell membrane. Here it is a key provided to understand more in detail about the layers present in the microorganism.
Moving on to the biochemical tests. The first biochemical test is Oxidase Test. Cytochrome containing organisms produce an intracellular oxidase enzyme. This oxidase enzyme catalyzes the oxidation of cytochrome C. Organisms which contain cytochrome C as a part of their respiratory chain are oxidase positive, and the ones which do not contain cytochrome C are oxidase negative. Oxidase positive bacteria possess cytochrome oxidase or indophenol oxidase and iron containing hemoprotein. Both of these catalyze the transport of electrons from donor compounds like NADH to electron acceptors, usually oxygen. The test reagent tetramethyl p-phenylenediamine dihydrochloride acts as an artificial electron acceptor for the enzyme oxidase. Hence, when cytochrome C oxidase enzyme is present, your reagent gets oxidized into indophenol, which is a dark blue purple compound, whereas when cytochrome C oxidase is absent, your reagent remains in its reduced form, which is colorless.
There are various ways to perform this test. One such way is wherein you dip a Wortmann filter paper 1 in the solution of the dye and then for like 13 seconds and then dry it. So these filter paper soaked in the dye are then used as the medium on which you will test your organism. So then you take a loopful of your organism and then streak on these filter papers, and if you develop a purple bluish color, then your organism is positive for oxidase enzyme, and if no color is developed, then your organism is negative, that is, it doesn't produce oxidase enzyme.
The next biochemical test is your catalase test. The enzyme catalase mediates the breakdown of hydrogen peroxide into oxygen and water. The presence of this enzyme in a bacterial isolate is evident when a small inoculum is introduced into hydrogen peroxide and rapid elaboration of oxygen bubble occurs in the form of effervescence. The lack of catalase is evident by a lack of effervescence or weak bubble production. By producing this enzyme, bacteria protect themselves from the lethal effect of hydrogen peroxide, which is accumulated as an end product of aerobic carbohydrate metabolism.
So herein we take drop of hydrogen peroxide on a glass slide, and then we take our inoculum So here you can see the presence of effervescence, which indicates that your organism is catalase positive, that is, it produces catalase enzyme, and if it doesn't, it won't produce effervescence, which you can see in this part, wherein it doesn't produce any effervescence, which means that your organism is catalase negative.
The third biochemical test is the Voges-Priscure test. Pyruvic acid, the pivotal compound in the fermentative degradation of glucose, is further metabolized through various metabolic pathways depending on the enzyme systems possessed by different bacteria. One such pathway produces acetyl methyl carbinol, that is acetone, a neutral reacting end product. If acetone is present, it is converted into diacetyl in the presence of alpha naphthol and a strong alkali like potassium hydroxide and atmospheric oxygen. The diacetyl produced reacts with guanidine containing compounds in your broth peptones and condense to form a pinkish red polymer.
Now for this Voges-Priscure test, you need
a 24-hour-old culture which is grown in a broth medium. Then you take 2 ml of the aliquot broth in a clean test tube, to which you add 5 drops of 5% alpha-naphthol and 2 drops of 40% KOH. Shake the tube well to aerate, and then if you find a pinkish red color develop in your tube, it is an indication of the presence of diacetyl, which in turn indicates the presence of acetyl methyl carbinol in your test organism. And if you do not observe a pinkish red color, and if the normal yellow color of your medium is retained, then it indicates that your test organism is negative for Voges Pritzker test, that is, it does not contain any acetyl methyl carbinol. The fourth biochemical test is citrate utilization test. Citrate agar is used to test the organism's ability to utilize citrate as a source of energy. The medium contains citrate as the sole source of carbon and inorganic ammonium salts as the sole source of nitrogen. Bacteria that can grow on this medium produce an enzyme, citrate permease, capable of converting citrate to pyruvate. Pyruvate can then enter the organism's metabolic cycle for the production of energy. The released carbon dioxide will subsequently react with water and sodium ion in the medium to produce sodium carbonate, an alkaline compound. This will raise the pH of the medium. To add to it, ammonium hydroxide produced on the utilization of ammonium salts also increase the pH. With an increase in the medium pH, bromothymol blue indicator changes its color from deep forest green to blue. Also, the presence of growth in the media is an indication of citrate utilization. For citrate utilization test, usually citrate agar slants are used, but for demonstration purposes, here we have used solutions. So your test organism is streaked on citrate agar slants and then incubated at respective temperatures for respective period of time, and then the color change in the slant is observed. So if you get a deep blue color on your slant, then it is an indication that your organism utilizes citrate as the sole source of carbon, and subsequently it results into an increase in the pH of your media, which converts bromothymol blue indicator from green color to blue color. And if the slant doesn't develop any blue color, it is an indication that your organism is negative for citrate utilization test, and it doesn't increase the pH of the media, and hence bromothymol blue indicator remains as it is, that is in its deep forest green color. The following key summarizes the positive and the negative results for all the 4 biochemical tests performed. The following sheet summarizes the observations for morphological characters and the results of biochemical tests that we have got for the 6 operational taxonomic units, that is the 6 bacteria taken into consideration. Now, let us compare these bacteria together using a similarity matrix, which will eventually help us to classify the organisms in a phylogenetic tree.
As we are done with all the characteristics, we move on to the similarity matrix. We have taken 2 species, A1 and A2. For every similar species, for every similar character, we will draw one line in the respective coordinate. So, for A1 and A2, the shape is bacillus and bacillus. That is, the character of both of them is similar. So, we draw one line in the respective matrix. So, A1 and A2. So, there is one line. Similarly, for size, the size aren't similar, so we won't draw any line. Endospore, no, yes and no. Similarly, it is not matching. Flagella, parenchymous and parenchymous. Therefore, we draw another line. Capsule, for both yes, another line. So the gram nature of one is positive while the another is negative. It is then similar. Oxidase, A1 is positive while A2 is negative. For the catalase test, both are positive. So we draw another line. Voges Pritzker So, as we see, there are 6 lines, that is, it matches 60%
with each.
Similarly, we can calculate for every other organism in similarity matrix. So, the similarity matrix for other bacterial species was calculated as we have talked earlier. This was the resultant matrix. So, in which we can see that A5 and the A6 bacterial species matches the most, that is the 70%. We can start drawing our phylogenetic tree. So, A5 and A6. So, here we can see that the A5 and A6 are more similar. Now, we compare these two microorganisms with other bacterial species. The rest of the matrix remains the same. Now, we calculate the coefficient for A5 and A6 with A1. For A1, A5 is 20. For A6, it is also 20. So, we will add 20 and calculate the average, which results into 20. So, it shows 20% similarity with A1. Similarly, for A2, it is 30. For A3, it is, and for A4, the A5 is 60, and for A6, it is 50. By finding out its average, it's 55% similar to A5 and A6. Now, here we can see that A5 and A6 are more similar with A4. So, we can draw one more branching and show that A4 is similar to A5 and A6.
Now A4 is most similar to A5 and A6. So now the next step goes, we will compare A4 and A5 and A6. So this whole phylogenetic matrix with the other organisms, that is A1, A2 and A3. So now let's start with the first organism, that is A1. So A1, when compared with A4, we get 30% similarity. Then when A1 is compared with A5 and A6, we get 20% similarity. So now, we will calculate its average, which comes out to be 25. So your A1, when compared with A4 and A5 and A6, it comes out to be 25. Now next organism is your A2. A2, when compared with A4, we have 30, and A2 when compared with A5 and A6, again we have 30. So the average of both A4 and A5 and A6 comes out to be 30. Moving on, the only organism left is now A3. So now when A3 compared to A4, we get 50 as the similarity percentage. So we'll write here 50. Then when your A3 is compared with A5 and A6 both, the similarity that we get is 40%. So we will write here 40%. And now we will calculate the average, which is 45. So moving on, here we will write 45. So your A3 is the most similar to your A4, A5 and A6, as the similarity percentage is the most when compared with A1 and A2. So now, again, another branch will result from this phylogenetic tree, which will evince your A3. previous similarity matrix, that is calculate the average. So this averages out to be 27.5%, which we will write here. And similarly for A2, when we calculate the average of 50 and 30, 50 plus 30, 80 divided by 2 is 40. So your A2 is 40% similar to all these four organisms. And another thing which we can notice from this matrix is your A1 and A2 are 60% similar with each other. So there can be another branch of two similar organisms, which is A1 and A2. A1. So your A1 and A2 are 60% similar to each other, which is more than a 40% similarity, and hence these two organisms have been clubbed here together, and now your A1 and A2 have a similarity of your A2 has similarity of 40% with your rest of the phylogenetic tree, and hence you can draw another branch from your phylogenetic tree, which will take you to your A1 and A2. So the final phylogenetic tree that will result is this.
Based on the observations followed by the construction of similarity matrix and its calculations, the organisms have now been classified, and this is how the final phylogenetic tree looks like. The following are the merits of numerical taxonomy. The following are the demerits of numerical taxonomy. Thank you.